Welcome student. Today we will see structure of pollen grain. This is a pollen grain, paragakan. Structure of pollen grain. Now one science is there, that is a palynology. Palynology means the science which deals with the study of pollen. We call it as a palynology. Now pollen grains, paragakan, we know that how this pollen grain formation takes place in anther lobe. From the sporogenous tissue, we call it as a microspore mother cell or pollen mother cell. By the process of meiosis cell division, microspore 4 tetrad 4 microspore formation takes place in tetrad condition, and it get differentiated into the structure. We call it as a pollen grain. Now this is structure, this one, this a pollen grain is nothing but a cell. It is a plant cell. Plant cell means in the cell there is a fluid which is present. That fluid we call it as a cytoplasm. Here cyto means a cell and this a plasm means a fluid. Means the fluid which is present in the cell, this one cytoplasm. And this cytoplasm surrounds with this structure. There is a structure, we call it as a plasma membrane. Plasma, again it is a fluid, and this one is the membrane. Means this is a plasma membrane. It is made up of fluid. It made from the fluid. It made from the cytoplasm. It is a cytoplasmic in origin, and it surrounds that fluid cytoplasm, that membrane, auron, we call it as a plasma membrane. These all structures, they are present in the plant cell. Now this pollen grain, it is unicellular. Uni means one. Cellular means it is made up of one cell. It is uninucleate. Uni means one. Nucleate means a nucleus. In this pollen grain, one nucleus is present, which is haploid in nature, consists of half chromosomes because it is formed by the process of meiosis cell division. Then this pollen grain, it is oval. In some species it is a spherical. In some species it is like this rounded. This pollen grain is a male reproductive body, measures from 10 to 200 mu. It is very small in size. 10 to 200 mu means these pollen grains are very small, minute, and only detect in the microscope. They are microscopic in nature. And these pollen grains, they are rich, in which rich nutrients is present in the peripheral cytoplasm. Peripheral cytoplasm, this one is the cytoplasm, and peripheral cytoplasm means in the mature pollen grain, vacuoles are present. And if big vacuoles are present here in this pollen grain, ultimately the cytoplasm, it shifted towards the periphery. The cytoplasm gets shifted towards the periphery, and the nucleus also move towards the periphery. Means nucleus, it do not present in the center. It is due to this vacuole. Then this pollen grain, it consists of EXINE and intine wall. This one is the EXINE wall, this one, and this one is the intine wall. It means this one plasma membrane covered with the two wall. That wall we call it as EXINE wall and intine wall. EXINE and intine wall, this is a word denoted by this scientist known as Frischchi. And this EXINE and intine wall, we call it as a Sporoderm. Sporo and derm. This one is the spore, microspore. Which one is the microspore? Cytoplasm. This is a haploid nucleus, vacuole, and these structures surround with this plasma membrane. This structure we call it as a microspore. And when this microspore surround with these two wall, EXINE and intine wall, we call it as a derm, sporoderm. Spore, this one is the spore, microspore, and derm means this EXINE and EXINE. This is the derm, means the skin of this pore. Sporoderm. The smallest pollen grain observed in case of this myosotis, it measures up to 10 mu in diameter, and largest pollen grain observed in case of this mirabilis, which measures up to 250 mu in diameter, like this. Now see the detailed structure of this pollen grain. Pollen grain, it consists of this wall, this one wall, we call it as EXINE, and this wall we call it as intine. EXINE, X means outer, X, in means what? Inner. EXINE and intine. EXINE, it is a thick, hard, and it is made up of Sporophyllonin, and intine, it is a thin, made up of cellulose and pectin, like this. Let us see this EXINE wall, pollen grain wall. It is made up of two walls. One is the EXINE and second one is the intine. This one EXINE and intine. Now EXINE is also made up of like these are two. Means this EXINE, it is made up of these are two again sub walls. We call it as EXINE, and second we call it as EXINE. EXINE and end EXINE are the parts of EXINE. And this EXINE also made up of like these three sub layers. One is the tectum, that is the outermost layer. Means this one is the outermost layer. We call it as a tectum. Beneath the tectum, next layer is there, that is the middle. We call it as a baculum. These EXINE get differentiated into like these layers. And the innermost layer, we call it as a foot layer. Like this, innermost layer, we call it as a foot layer. Means this EXINE get differentiated into these three layers: tectum, baculum, and foot layer. Tectum, outermost layer; baculum, middle layer; and foot layer, that is the inner layer. Forms this EXINE. It is a part of what? EXINE. And second, we call it as what? Intine. So this EXINE and intine, in the formation of these pollen grain, it is secreted by the nutritive tissue which is present in the microsporangium. That tissue we call it as tapetal cells or tapetum. And in that tapetum, in the cytoplasm of tapetum, special bodies are formed. We call it as a yellowish body. And yellowish body form these EXINE as well as this intine, like this. And when EXINE and intine get formed, surround these microspore, then that structure we call it as pollen grain. Then that structure we call it as what? Pollen grain. So let us see the details of the EXINE, intine, then these germ pore, and the pollen kit. EXINE, this one, EXINE, this one, outer wall of this pollen grain. EXINE, it is made up of Sporophyllonin. EXINE, it is made up of what? Sporophyllonin. Means this EXINE, it is made up of Sporophyllonin. And what is Sporophyllonin? Sporophyllonin, it is made up of fatty acid. This Sporophyllonin form on this EXINE, due to which it become thick, this structure become hard, and due to this Sporophyllonin, this EXINE is ornamented. Ornamented means it shows sculpturing. Sculpturing means it shows many like these different like these structures on these EXINE. Like these structures are seen on the EXINE, like this. Different type of like these sculptures are seen on the EXINE, like this. And it is made up of Sporophyllonin. We call it as ornamented. Now these Sporophyllonin, it exhibit these Sporophyllonin which forms such a type of sculpturing on the pollen grain, exhibit a fascinating array of pattern and different design on the pollen grain. And due to which we can determine the species of a specific a specific plant. Then Sporophyllonin, it is a most resistant organic material. Sporophyllonin, it is most resistant organic material, withstand high temperature and strong acid and alkali. There is no enzyme till this date. There is no enzyme which degrade the Sporophyllonin so far. Such a strong material is formed in the nature. Now EXINE and this Sporophyllonin made from the yellowish body. This yellowish body, they are present in that we call it as a tapetal cell. In that tapetal cell cytoplasm, yellowish body formation takes place. Yellowish body transform into sporogenous tissue, and when the pollen grain formation process is going on from this yellowish body, these all Sporophyllonin and these EXINE formation takes place in case of the pollen grain. Pollen grain, it is a well preserved as a fossil. Means this is a small microscopic structure. It get well preserved in the fossils also, because the Sporophyllonin, it is very protective material, and due to that Sporophyllonin we can get the fossils of pollen grain also. The study of these hard remains in the what that earth, we call it as what? Fossil. Now this EXINE wall, this EXINE wall is discontinuous. Means this EXINE wall here, it is discontinuous like this. It is not a continuous wall. The next wall of this pollen grain, we call it as intine. This one intine. This intine, it is made up of cellulose and pectin. Then this intine, it is the inner sporoderm of pollen grain. It is a inner sporoderm. Derm means skin, inner skin of this pollen grain. Then it is a thin and continuous, surrounds the plasma membrane of microspore. It is a thin, this one, it is a thin, and it is a continuous, and it surrounds these plasma membrane. These are what? Microspore. These intine contribute in the formation of pollen tube. When the pollen tube formation takes place, when this is a mature pollen grain, it get a transfer from mature anther lobe to the receptive stigma of another flower of the same species. It absorbs water from the stigma of that stigma of gynoecium, and the volume of these cytoplasm get increased, due to which it exerts a particular pressure on the wall of these plasma membrane, and due to these hydrostatic pressure from these germ pore, these we call it as intine, which is made up of cellulose and pectin. These intine come out like this, and like this a tube formation takes place, and this is a tube, we call it as pollen tube. So like this, the pollen tube get emerge out through these pores. That pores we call it as a germ pore. Here germ, germination, pores means microscopic small opening. We call it as a germ pore. Then germ pore, where the Sporophyllonin is absent. This is a germ pore present where the Sporophyllonin is absent. Means Sporophyllonin do not present here, due to which here pore formation takes place. That pore we call it as a germ pore. And through this germ pore, intine come out in the form of a pollen tube. Now in case of dicot plant, in case of what? In case of a dicot plant, Dvibijapatri plant. In case of a dicot plant, see here, this plant is a dicot plant. This plant we call it as the Ticoma stans. This one. In case of this plant, in case of dicot plant, 3 germ pores are present in the pollen grain. 1, 2, and 3 germ pores are present in the pollen grain of this plant. Second example, see this one. This one. This one is the examples of Vinca rosea. It is also dicot plant. Means in this dicot plant, in the pollen grain, 3 germ pores are present. 1, 2 and 3. Next examples here. This one. We call it as Aza directa indica, neem. In this plant also, the pollen grain which is formed in that pollen grain, 1, 2, 3 germ pores are present. But in case of monocot, for example, this one is the monocot, and this is a plant, this one, we call it as a typha. What is it? Typha. And in case of these monocot plant, typha or grass or bamboo or like this type of plant, monocot plant, jawar, bajra, in case of this monocot plant, only one pore is present. Pollen grain is there, but in the pollen grain, only one pore is present here. Single pore is present. One single germ pore is present in case of what? Monocot plants. Then come to the pollen kit. Pollen kit means in insect pollinated plant, not in wind pollinated plant, not in case of water pollinated plant, but in case of insect pollinated plants, a oily thick viscous layer which is made up of lipid and carotenoid, due to which pollen adhere with each other. Pollen adhere with each other and it forms a kit. That a kit we call it as a pollen kit. Number of pollen grain get adhere with each other due to these lipid and this carotenoid, that is a viscous layer, or due to that oily layer. Many pollen grain get adhere with each other, and that many pollen grains means, for example, hundreds of pollen grain which is easily visible by our naked eye in case of Hibiscus rosa sinensis, just one. If we put that Hibiscus rosa sinensis anthers on our skin, we can easily detect that yellow powder which is present on our skin, which is stick on our skin. Means one dot of that yellow powder, it means we can see by our naked eye. It means in that one visible dot, hundreds of pollen grains are there. That kit we call it as what? Pollen kit. That we call it as a pollen kit. So due to these oily and this viscous layer which is made up of lipids and these carotenoids, when the insect visit here on this flower to suck the nectar from this flower, at that time from the anther, due to this pollen kit, due to this oily and this viscous layer made up of lipids and carotenoid, due to which the pollen grain get adhere to the skin of that insect, to the wing of that insect, to the legs of that insect, and pollen grain get a transfer from one flower to another flower with the help of this insect. We call it as entomophily. So the pollen grain get adhere to the body of the insect. Now next function of this pollen kit is, this pollen kit attract the insect, because that pollen grain get easily detected by the insect, and that insect they attract towards the pollen grain or attract towards that flowers. Then these pollen kit protect the pollen grain from ultraviolet rays. Means they has a capacity to protect that pollen grain from the ultraviolet rays, due to which mutation do not take place in that small tiny particles, that we call it as a pollen grain. Nowadays pollen syrup formation takes place. Some companies are there, they are also preparing that pollen tablets, because in this cytoplasm of pollen grain, this cytoplasm is rich in carbohydrate, rich in protein, rich in water, and they assume that these pollen syrup or these pollen tablets, they improve their power in case of the athletes. Then nowadays pollen allergy also detected. There are some pollen respiratory disorders also there detected in the humans. One plant is with me. This is a plant, we call it as a parthenium. This one. This plant we call it as a parthenium. This one is the Congress grass. And here the pollen grain formation takes place here. We call it as a conidia. Large amount of conidia, large amount of these spores, tremendously they move from here and there. In this room also these pollen grains of these Parthenium is present, and there are some people, they are very sensitive these Parthenium pollen grain, and they shows the allergy. We call it as pollen allergy. And some individuals are there, they show the respiratory disorder due to these pollen grain. We call it as a pollen respiratory disorder. Then last point is there, pollen of Sergenia latheris. That pollen grain of Sergenia latheris. These pollen grains are poisonous in nature. In the cytoplasm, some poisonous contents are present. And these are plants only found in case of Brazil. And in the Brazil, honey collect these pollen grain. And if such pollen grains are present in the honey, then that honey, it becomes poisonous. There are so many deaths occur in the Brazil due to about the development of male gametophyte. Gametes, how the gametes formation takes place in the structure, we call it as a pollen grain. How these gametes get developed, these are two male gametes, how they develop, we will see here. Development of male gameto, these are gamete, these are the two male gamete, and phyte means plant structure, it is a plant cell. This one pollen grain, it is a plant cell, unicell structure. Now see here, one first slide, in this first slide here, tetrahedral type of pollen, tetrahedral types of these microspores, tetrad is present, is shown here in this first slide. In the second slide, how the pollen tube emerge out. See here, this one is the pollen tube, how the pollen tube emerge out from the pollen grain, it is shown. And in third slide, see here, the pattern of the Sporopollenin on the EXINE wall. This is a Sporopollenin, it is a protective material, and how the pattern of these Sporopollenin formation takes place on the EXINE. See here. And in the fourth slide, it shows the pollen tube germination here. How the pollen tube germinates. These all are the pollen tube in this fourth slide. Now the development of male gametophyte. This one, that is the structure of pollen grain. We know that structurally pollen grain, it consists of outer wall known as EXINE, inner wall, that is the intine. Then beneath the intine here membrane is present, and membrane we call it as a plasma membrane. In these beneath this plasma membrane here the cytoplasm which is present. In the cytoplasm, like this nucleus is present. This nucleus is haploid in nature, and previously there is no vacuole, but as the pollen grain mature, vacuole formation takes place, and due to which the nucleus shifted towards the periphery, and cytoplasm also arranged here in the peripheral region like this, due to these big vacuoles. This one, that is the cytoplasm present towards the periphery of these pollen grain, in the pollen grain like this. Now the development of these male gametophyte, development of a male gametophyte is endosporic or in situ. Means development of these two male gametophyte, these are male gametophyte, it is endosporic in nature. Endo means inside the cell, means inside the cell here events takes place. And lastly, like these two male gamete formation takes place, due to which development we call it as endosporic, or it is called as in situ, means inside the cell. Then male gamete, by this a male gamete which is there, this one, this is the male gamete formation takes place by two mitotic division. It means this one, the pollen grain, when there is a pollen grain present inside the anther lobe, in each anther lobe two microsporangia are present, and inside this microsporangia, in one microsporangium, that pollen sac is present. Inside the pollen sac, like this minute teeny small unicellular uninucleate, like this microscopic many uncountable pollen grains formation takes place. In that pollen grain, this nucleus is present, and this formation of male gametophyte, or this development of male gametophyte get differentiated into that two events. One we call it as before and after pollination events. Before pollination events is going on in the pollen grain, and after pollination there are some events which are going on in these pollen grain. Which are the before pollinated events? Before pollination, in the pollen sac, what happened? These are pollen grain which consist of these functional nucleus. It undergoes in first mitotic cell division. Mitosis is the equational cell division in which the chromosome number remains as it is. Means this nucleus, it undergoes a mitotic, it shows a mitotic cell division, and this nucleus divided into two nucleus like this. This nucleus get divided into two nucleus like this. And when this nucleus get divided into two nucleus, here a cell wall formation takes place. Here a cell wall formation takes place like this. Now when the cell wall formation takes place, due to this cell wall, two unequal cells formation takes place. Due to these cell wall, like these two unequal cell formation takes place within the pollen grain. The bigger cell in which more cytoplasm is present, this bigger cell we call it as a vegetative cell, or it is also known as a tube cell. And the smaller cell which is there, this one, this is a smaller cell which is a spindle shape. This is a smaller cell, we call it as a generative cell. Janan koshika, Janana cell, we call it as a generative cell, means it form male gamete in future. The bigger cell we call it as a vegetative cell, or this bigger cell we call it as a tube cell. And in this vegetative cell or the tube cell, the nucleus which is present, that nucleus we call it as a vegetative nucleus. And in the generative cell, the nucleus which is present, that nucleus we call it as a generative nucleus. Now the nucleus which is present in this bigger cell, big cell, known as a vegetative cell or tube cell, this nucleus is irregular. The shape of this nucleus is irregular. But the nucleus which is present here in this generative cell, this nucleus is a functional cell. This nucleus is a functional nucleus, and this nucleus again divided. It shows a second mitotic division, and it forms like this two male gamete. Now the pre, or we call it as a before pollination events. These all are the before pollination events. Now the pollen grain, it appears a two cell structure. One cell, bigger cell, vegetative cell or tube cell, and second cell, this one we call it as a generative cell or generative cell like this. Like these are two cells are there. Now see here, this generative cell is covered with this. Actually, this generative cell is very small cell. It is a spindle shape. It consists of a spindle shape structure. Here cell wall is there, but this cell wall do not made up of cellulose. The cell wall of this generative cell, it is made up of this material, we call it as a callose, and just it adhere to the plasma membrane of this bigger cell, that is a vegetative cell. In next stage, this generative cell, it get separate from this plasma membrane. Due to this material, we call it as a callose, and it float here in the cytoplasm of this vegetative cell or the bigger cell, and due to which it forms like these are two male gametes here. Now see here, when in the majority of plant, means in the 60% plants, angiospermic plant, pollen grain, when the anther lobe mature, the anther lobe dehisce, and when the anther lobe get dehisce, these pollen grain get a release. In 60% angiospermic plant, pollen grain get a release at a two-cell structure. Means majority of plant they release pollen grain of a two-cell structure. But there are some plants, means the remaining 40% plants are there, they release these pollen grain of a three-cell structure. How this is three-cell structure formation takes place? See here. What happened? These are generative, these are cell which is there, the generative cell. It consists of these cell wall, but these cell wall do not made up of what? Cellulose. The cell wall of this generative cell, it is made up of this material, we call it as a callose. Callose is nothing but a carbohydrate, means it is made up of what? Carbohydrate. Now what happened? Before pollination, in 40% plant, what happened? These are generative cell, it divide mitotically. And when it divide mitotically, these generative cells divide mitotically, and it forms like these are two nucleus. One and this one second nucleus. The nucleus divide mitotically, and it surrounds by this material, we call it as a callose, like this. It surrounds with this material, we call it as a callose. So this one is the callose, and this one we call it as a generative nucleus. This one generative nucleus, this one is the generative nucleus. Now this one is the one cell, this one is the second cell, and this one is the third cell. Near about 40% angiospermic plant, they release pollen grain at such a three-cell structure. 1, 2, and like these are three cell. But a 60% angiospermic plant on the earth, they release a two-cell structure, one cell bigger cell, that is a vegetative cell or tube cell, and second here that is spindle shaped cell, that cell we call it as what? Generative cell. These all are the pre-pollination events. Now when the pollen grain released from the mature anther lobe, now this pollen grain it transfer by various agencies, and transfer of this pollen grain from mature anther lobe to the receptive stigma of same flower or another flower bearing on same plant or another plant. On the stigma of that gynoecium, we call it as a pollination. Now this pollen grain move towards the stigma. This one we call it as a stigma. Then this one is the long style, and this one is the bulbous ovary. This one we will see the details of this gynoecium, ovule, and pollination and all in next video. Now these pollen grain of two-cell structure in near about 60% plant, it get a transfer here by various agencies, and they adhere here to these stigma. This one is the stigma. This one style, and this one is the ovary, bulbous ovary, in which like this ovule is present. Now when the pollen grain get deposited here on the stigma of the same species, then these pollen grain absorb water or sugary material from these stigma, and when the pollen grain absorb water through the germ pore, or we call it as a germ furrow, water move inside these pollen grain, due to which the size of the pollen grain get increased. And when the size of the pollen grain get increased, the water exerts a pressure on the wall of enzyme. This one is the intine wall, this one. So this intine wall, it comes out here like this. Intine wall, it comes out like this through these germ pore or germ furrow like this. This intine wall comes out, and these intine walls, it comes out, it is due to one pressure. That pressure we call it as what? Hydrostatic pressure, or we call it as the turgor pressure, which is created by the water, and this excess water comes here in this cytoplasm due to the absorption of this water from the stigma, and due to which these pollen tube it emerges out. This tube we call it as a pollen tube. In the formation of these pollen tube, these tube cell, bigger cell, bigger cell play important role, due to which these pollen tube get formed. Here the pollen tube get formed like this. And when the pollen tube get formed, the vegetative nucleus, this one we call it as a tube cell. This nucleus also we call it as a tube nucleus. This one is the tube cell. The nucleus we also call it as what? Tube nucleus. The tube nucleus first move here, and it comes at the tip of what is a pollen tube. These structure we call it as a tube nucleus, or this we call it as a vegetative nucleus. Means first this structure come here in the pollen tube, and then along with the cytoplasm move here, and along the movement of cytoplasm, first this tube nucleus or vegetative nucleus move, and tube nucleus comes here at the tip of this pollen tube, and in future it get disintegrate, it get dissolved. And the functional cell which is present here, that functional cell, this one, that functional cell, that is a spindle type, this is a cell. This is a cell we call it as a generative cell. Now this is a generative cell. Which is present here, which is separate, which get separate from this plasma membrane due to these callose. Now these generative cell shift here in these pollen tube, and here second mitotic division takes place. Second mitotic division takes place here. These events we call it as a after pollination events. After pollination events, these pollen tube formation takes place. In this pollen tube, cytoplasm comes. In the cytoplasm, first this tube nucleus or vegetative nucleus comes. Then along the cytoplasm, this generative cell comes here. In the generative cell, here second mitotic cell division takes place, and due to which lastly, like these two male gametes formation takes place. Now see here, these two male gametes are stationary in nature. They are stationary in nature, means they do not move. Due to which here these pollen tube formation takes place, and this pollen tube move here and ultimately reach these these male gamete up to these egg like this. These all are the development of what? Male gametophyte. If you want to